Welcome to today's lecture on cancer biology and oncogenes. We will explore how genetic alterations lead to abnormal cell growth and the development of cancer. Topics include cell cycle control, DNA damage, oncogenes, tumor suppressor genes, and therapeutic approaches. The cell cycle controls cell growth and division through phases such as G1, S, G2, and M. In cancer, mutations in cell cycle control genes disrupt the normal progression, causing uncontrolled cell division and tumor formation. Tumor suppressor genes are vital for regulating cell growth and preventing cancer. These genes produce proteins that maintain genomic stability, preventing tumor formation. Mutations or silencing of these genes result in uncontrolled cell growth and cancer development. Common tumor suppressor genes, like TP53, P53, BRCA1, and BRCA2, have been linked to increased risk of breast and ovarian cancer. Studying their role in carcinogenesis is crucial for developing targeted treatments and prevention methods. In cancer cells, DNA damage can occur from both external sources like UV radiation and chemical carcinogens, as well as internal sources like errors during DNA replication. Cancer cells often have inefficient DNA repair mechanisms, leading to the accumulation of DNA damage. DNA repair pathways, such as base excision repair, nucleotide excision repair, and homologous recombination, are essential for preventing mutations caused by DNA damage. When these repair pathways are disrupted, it can result in genomic instability, which is commonly observed in cancer cells. Oncogenes are specific genes that, when activated, promote cell growth and division. These genes are usually involved in signaling pathways that regulate cell proliferation, differentiation, and survival. Activation of oncogenes can occur through various mechanisms, such as gene amplifications, chromosomal rearrangements, or point mutations. Once activated, oncogenes can drive abnormal cell growth and contribute to the initiation and progression of cancer. Examples of well-known oncogenes include RAS, HER2, and MYC. Proto-oncogenes are normal genes that, when mutated or altered, become oncogenes. These transformed genes disrupt normal cell processes and promote uncontrolled cell growth, leading to cancer. Identifying how this transformation occurs is crucial for developing cancer treatments. Signal transduction pathways play a central role in controlling cellular responses to various extracellular signals. Dysregulation of these pathways can lead to uncontrolled cell growth, survival, angiogenesis, and metastasis in cancer. Examples of well-known signaling pathways involved in cancer progression include the MOP/ERK pathway, PEEK/AKT/mTOR pathway, and the WNT/beta-catenin pathway. These pathways integrate extracellular signals and transmit them to the nucleus, resulting in changes in gene expression and cellular behavior. In addition to genetic mutations, cancer development is often accompanied by epigenetic alterations. Epigenetic changes refer to modifications in gene expression that do not involve alterations in the DNA sequence itself. Epigenetic modifications, such as DNA methylation and histone modifications, can contribute to the silencing of tumor suppressor genes or the activation of oncogenes. These alterations play a critical role in the initiation and progression of cancer and can be potential targets for therapeutic interventions. The tumor microenvironment, composed of cancer cells, immune cells, fibroblasts, and blood vessels, plays a crucial role in cancer progression. Cell interactions in this environment influence tumor growth, invasion, and metastasis, with immune cells having a dual effect. Detailed understanding of these dynamics is essential for developing effective treatment strategies. Cancer is characterized by several hallmarks, which collectively contribute to its development and progression. These hallmarks include sustained proliferative signaling, evasion of growth suppressors, resistance to cell death, induction of angiogenesis, ability to invade and metastasize, reprogramming of energy metabolism, evasion of immune surveillance, and genomic instability. Understanding these hallmarks is crucial for a comprehensive understanding of cancer biology and for the development of targeted therapies that address these specific characteristics of cancer cells. Molecular markers are specific molecules or genetic alterations that can be used to detect, diagnose, and determine the prognosis of cancer. These markers can include specific gene mutations, gene expression patterns, protein expression levels, or alterations in DNA or RNA. Molecular markers play a critical role in early cancer detection, monitoring treatment response, and predicting patient outcomes. Advances in molecular marker identification and detection technologies have significantly enhanced our ability to diagnose and manage cancer more effectively. Targeted therapies disrupt key molecules or signaling pathways in cancer cells, reducing toxicity and improving outcomes. Drugs like tyrosine kinase inhibitors and monoclonal antibodies inhibit oncogenes, transforming cancer treatment and benefiting patients. In cancer research, cutting-edge technologies like next-generation sequencing allow for the in-depth analysis of cancer genomes, transcriptomes, and epigenomes. This helps uncover the specific molecular changes responsible for cancer growth and allows for more personalized treatment strategies, known as precision medicine. By understanding each patient's unique genetic alterations, doctors can select targeted therapies that have a higher chance of success. In conclusion, our understanding of cancer biology has been greatly advanced by the field of molecular biology. By examining cell cycle control, tumor suppressor genes, DNA repair, and oncogenes, we have made significant strides in cancer research and treatment. Exciting developments in genomics hold promise for improved diagnosis and personalized therapies.